So I won't lie to you, I'm not a stem cell biologist, I'm a developmental biologist. So the talk today will be more developmental biology based. Look at polarity and the context. In the context of renal development, actually. Okay. Oh.
Let's try this again. Let's get this lecture right here.
That one. Yes.
Oh, come on now.
A lot of slideshows.
There we go. Up and down. Right.
No.
Oh, that's not
Okay.
me, no.
That's
There's
not
someone
you.
else.
Oh, no wonder.
That's the last one.
Well, let's get you out of there.
That's me.
There you go. How about right here?
I have a couple
Let's-
movies I
let's-
might not play, but
Let's
it's
close-
alright.
It's all right. Let's close this one out. And there you go, right there, slideshow. Okay, we know, whatever. Up and down right here.
Thank you. Okay, so I studied tubulogenesis in the zebrafish pronephros. On this title slide here we have a cadherin 17 line that labels all the tubule cells of the zebrafish nephron. The nephron is the simplest functional unit of the kidney. And that's what really is going to be the focus of my talk today, is the kidney. So I should start with 1 in 9 Americans, or 26 million people in the United States, or 300 million people worldwide, are afflicted with some form of kidney dysfunction. This represents the 12th leading cause of death worldwide. The simplest functional unit of the kidney is the nephron. The nephron is composed of 3 major parts. It's got a blood filter called the glomerulus. It's composed of specialized cells called podocytes. And then a highly specialized epithelial tubule will be the major portion of my talk today, and then collecting duct that excretes waste. And then majority of pathologists in the nephron look at the kidney as a whole. Again, those hundreds, a human kidney is about the size of your clenched fist, and it's composed of hundreds of thousands, if not millions of nephrons. And so for a human nephrologist, it's very difficult to isolate any single nephron. But more so, they look at the kidney as a whole, and they isolate these fluid-filled cysts for polycystic kidney disease. And again, it's very difficult to see in this healthy kidney a single tubule, how it develops. However, the zebrafish offers us an amazing simplified model system. It has 2 nephrons during early development. So you can really simplify this mechanism by 10-folds of magnitude. And again, the zebrafish offers us, but the same intermediate mesoderm that develops the human kidney also develops the zebrafish kidney. And it forms by 24 hours post-fertilization, and it's functional by 48 hours post-fertilization. So zebrafish has a ton of other attributes that are really amazing. It develops ex utero, it's translucent, so you can actually see organogenesis processes happening under a simple dissecting microscope. It allows for a simple forward genetic microscopy, forward genetics, transgenics, and even chemical genetics. You can add molecules to a dish and look at how they affect the nephron development. So for my talk today, I'm going to focus on this organogenesis process here and how a nephron develops. So importantly, I think it's really important to state that the segments of the nephron are highly conserved between zebrafish and mammals. All of the segments of the nephron have the same solute transporters between zebrafish and humans. So the same solutes that are absorbed between zebrafish are also absorbed between humans. It's extremely highly conserved, and I'll add more evidence to that in a second. And it's my personal understanding that if you can understand development, you'll be better able to understand disease. And I'll get to that in a second too. So when I entered a lab about 5 years ago, I wrote a review, and we looked at the development of a zebrafish nephron. It's been studied for more than 20 years. We know how the zebrafish nephron is induced, how it undergoes morphogenic processes, and all these processes that undergo to form those different segments of the nephron. However, one aspect we did not understand was this cell state transition. When these cells of a mesenchyme underwent this cell state transition from a mesenchymal cell population to form that epithelial cell tubule with a lumen at its apical center. That's really what I wanted to understand. I want to understand this cell state transition, and that's really a principal concept of developmental biology and stem cell biology, and I'll get into that in a second too. So for my talk today, I'm going to focus on these first three. I'm not even going to get to the second aim here at all. So for the formation of a nephron, how does it form? So at early developmental time points, when I looked at using whole mount in situ hybridization, this looks at gene transcription profiles. We can use this to label all the cells of the intermediate mesoderm. We can watch them undergo this mesenchymal to epithelial cell transition to acquire this lumen at their apical center. We can watch this mesenchymal to epithelial cell transition occur in real time. And I wanted to identify the precise time this occurred. So I did this by, there's a gold standard in the zebrafish. We can double label somites, which are a developmental time course, by, and it's red staining here. Look at somites by developmental time course. And then we can label all the cells of the nephron by this purple stain, this cadherin stain. I can embed them into a plasticizing agent and then take histological sections through the cells and look at time course of assays. So to give you a rough schematic, what you'll be looking at here, again, my tissue of interest will be this intermediate mesoderm that lies just dorsal to the yolk sac extension and just ventral to the praxial mesoderm of those somites. And we can see this here again, and this intermediate mesoderm here, and I develop a transgenic line that labels all the cells of the intermediate mesoderm in green. And for scale purposes, this scale bar is 5 microns. So this is a very small area. This is 25 to 30 microns. We're looking at a very small. It fits in the eye of the Roosevelt on the dime. So we're looking at a very small area. This is similar to the human nephron as well. So in early histological observations at proximal, central, and distal regions, I saw this cluster of about 8 to 10 cells. And at later stages, I saw it become more circumferential in appearance, became more circumferential. And then at 20 somites, very importantly, I saw the first appearance of a lumen, and it was at proximal, central, and distal regions. And then as development proceeded, this lumen gained in diameter. I know this is a convergence and extension type mechanism. We look at tight junction formation. This also had the same delineation of time points as this lumen acquisition at this time point here. So to kind of summarize this data, or to give you a more full point of this, I don't want to use tubulogenesis and mesenchymal to epithelial cell transition synonymously. There are different processes, but one process that overlaps amongst the two of them is the acquisition of polarity, or the partitioning of proteins into apical and basolateral domains. And so one polarity complex that's known to do this is the ternary polarity complex, initially identified in the C. elegans embryo, and it's atypical protein kinase C, par 3 for partitioning defective 3, and par 6. And this localizes at an apical domain, also with actin, and it restricts the basolateral proteins such as sodium potassium ATPase, which is necessary to establish an ionic gradient and to establish tight junctions between proteins, so cell-to-cell contacts. And so sodium potassium, atypical protein kinase C is extremely highly conserved between the zebrafish and humans. And in fact, if you can see here, it's 88% identical between zebrafish and humans. The amino acids are 88% identical. It has 3 major protein domains. It has a PB1 domain, which is known to interact with par 6. It has a ceramide binding domain, which is known to interact with the plasma membrane or phospholipids. And it's a serine threonine kinase, which has more than 60 known downstream targets, one of which is the ezrin, rhodixin, moisin proteins, which I'll get into in a minute. So I wondered how was APKC propagating this polarity, and how is it working to propagate tubulogenesis? And there are two APKC isoforms in mammals, as there are in zebrafish. There's an iota and a zeta isoform. And so what I want to do is I want to study these two isoforms in the zebrafish, and we have a commercially available antibody that recognizes both isoforms. We have an antibody here. It's labeled in cyan. The movie's not going to play, I guess. But this antibody labels the apical isoform, labels the apical domain of the pronephros. We have that cadherin 17 line that labels all the cells in the intermediate mesoderm, and we have a red label here that labels actin and cytoskeleton, which localize to cell-to-cell contacts, and the nuclei is labeled by DAPI. So in time course analysis, we realized that APKC was localizing to the apical surface prior to the formation of a lumen. Now, if we go back to literature, Keith Mostev's group in murine canine kidney cell lines also found this in vivo. Now, we're the first to find it he found it in vitro. We're the first to find it in vivo in a vertebrate organism. This localized again into the apical membrane initiation site prior to the formation of a lumen. Now, once a lumen is formed, it continually encompasses the lumen and is there present throughout the development and into the adult mesonephros. Actin shows a very similar localization. It's very punctate, presumably the site of cell-to-cell contacts, and again becomes more band-like as development proceeds, but is still the site of cell-to-cell contacts. Now, sodium potassium ATPase, again, that molecule that's necessary to establish cell-to-cell contacts, is not typically basilarly localized at early development time points, but it does not overlap with sodium potassium ATPase, but at later time points, it is basilarly localized. So what I want to drive home the point here is that wild-type mesenchymal progenitors undergo the cell-state transition by a convergence and extension mechanism, and they take on an apical membrane initiation site to propagate proteins into an apical and basolateral domains, which we've all learned about in developmental biology, to form this apical lumen at the center of a tubule. But I really wanted to get the point home here is that what is polarity's role in all of this? And that's really what my novel finding came in. So the zebrafish offers us numerous mechanisms to knock down gene function. There's CRISPR-Cas, talons. I use morpholino oligonucleotides. And as I mentioned, there's two isoforms of APKC, an iota and a zeta isoform. So when I started my research, I wondered if there's redundancy between the two isoforms. There is redundancy between the two isoforms, and I'll show you that here. So when I knock out zeta alone, I get a pretty wild-type phenotype. This corresponds to the mouse knockout. The mouse knockout of zeta is adult viable. It only presents immunological defects later in life. However, the iota knockout gives me about a 40% severe phenotype. Now, the severe phenotype correlates with curved body axis, pericardial edema, patchy eye pigmentation, and again, we get a 40% severe phenotype in the iodomorphin, and this corresponds to the iodomorphin in the mouse model. If you knock out the iota in the mouse, it's embryonic lethal. You don't get any viable adults. Now, if I knock down both isoforms of my zebrafish, I get a more than 90% severe phenotype in my double-morphin embryos. And to give further evidence to this, we have that antibody that localizes to the apical surface, APKC, and again, in each of the single morphin embryos, we get some level of APKC at the apical surface, but in the double morphins, we do get a total loss of APKC and double morphin embryos. Now again, phosphorylated ezronidixamucin in the control also localizes the apical surface, and each of my single morphins, maybe it's hard to see here, but you get some level of phosphorylated ezronidixamucin at the apical surface, but it's totally lost in the double morphin embryos. Now I'll take a short break to let you look at this data. It's surprising to me. In my double morphin embryos, I did get a lumen formed in these double polarity deficient embryos. So personally I thought that polarity is going to be necessary to propagate a lumen. That was not the case. However, this is a 36-hour time point. So I wondered what was the earliest time point that a lumen was formed? How was tubulogenesis being propagated? So we took a time course of embryos, and at early time points I saw this disruption of sodium potassium ATPase. I saw a disruption of actin. I did not see the proper apical membrane initiation site being formed. However, at 36 hours, I did see a lumen being formed, and I did see an extracellular matrix being deposited laminin. So these tubules do eventually become formed, however, be it very much smaller than their control wild types. So that was interesting to me. So to summarize this data, in wild-type embryos, again, we have this wild-type embryo undergoing a convergence and extension mechanism, undertaking an apical membrane initiation site to propagate a lumen. In polarity-deficient embryos, we have the disruption of this, and a lumen is eventually formed, albeit slower, about 12 hours slower, in the zebrafish pronephros. So really the novel finding came when I looked at gene expression profiles, and we do this by whole mount in situ hybridization in the zebrafish embryo. And when I did this, so in a wild-type embryo, maybe you heard earlier by Dr. Romagnani, PAX2A is expressed in scattered cells in the next segment and just in scattered cells of the tubules. And it's exclusively expressed from the podocytes of the glomerulus region, where WT1, the Wilms tumor, is expressed. So this Wilm tumor is expressed in just the glomerulus, that blood-filtering organ of the renal origin. And one downstream factor of WT1 is podocalixin. And about 15 years ago, Majumdar et al. Out of Ian Drummond's lab identified a PAX2A mutant, and he found this PAX2A mutant extended this field of WT1 expression and from a wild type where it's only expressed in the podocytes to this neck region or tubule cells of the zebrafish pronephros. Now, my novel finding came here when I looked at my polarity deficiency, and the double polarity deficiency, and only in the double polarity deficient embryos did I see the ectopic expression amongst all of the tubule cells of the zebrafish pronephros, and every single cell of the pronephros was expressing PAX2A and Wilms tumor suppressor and the downstream transcription factor protocalyxin in all these cells of the zebrafish pronephros. And I know this is to be true because by co-labeling with cadherin 17, these cells are co-labeling with cadherin 17. And this is not the case in the single morphin embryos. So this is a pretty striking finding. And I wondered I went back to the literature and what it meant for a human. Now, if you look at human patients, for Wilms tumor, it's a terrible disease, actually. And prior to 15 years of age, if you have Wilms tumor suppressor, as the name kind of denotes, Wilms tumor suppressor is exactly that. It suppresses tumors in early adolescence, prior to 15 years of age. So if you have this expressed in your tubule cells, you're not in good health. Actually, it's a terrible disease, and it has terrible morbidity as well. Similarly, PAX2A is also a biomarker for renal cell carcinoma, which is the most common cancer of the kidneys. And here in a wild-type patient, we can see that by PAX2A, there's very little, if any, expression of PAX2A. However, in a renal cell carcinoma patient, all these cells, almost every cell, is marked by PAX2A. And this is a terrible diagnosis for a renal patient. Again, this is the 12th leading cause of death worldwide. This is awful. So we in the renal community wonder how we might be able to rescue this phenotype, how we can alleviate this population of people afflicted with this cancerous phenotype. And we tried a number of different things. We tried, I can't even mention how many different things we tried. But one thing that we did try that did work, if we take away that paired homeobox transcription factor, PAX2A, in a polarity deficient background, we can rescue that ectopic expression from the tubule cells of the distal region of the nephron. Now, albeit, so again, in control embryos, we get this expression of WT1A in the podocytes, the glomerulus of the nephron, that blood filter, and we get PAX2A in a mutually exclusive domain. And again, in our polarity deficient embryos, we get this ectopic misexpression. And then in the triple morphant knockdown, we can take away that ectopic expression from these distal tubule cells of the nephron. And it looks very reminiscent to what Majumdar saw 15 years ago. Where he saw this expansion of this distal expression domain in just this neck region here, where we determine a neck region. And I can statistically prove this rescue in bar graphs here. We can transverse this high ectopic expression to just this neck ectopic expression as shown here. And then interestingly, if you look at this very closely, this little panel here, which I really should have blown up, this little panel here, this cadherin 17, these cells are specified to become renal progenitors. However, those renal progenitors that are ectopically expressing protocalyxin and WT1 have a very low expression of WT1 in the PAX2A rescue. So these cells are really kind of fighting to figure out what their identity is, what epithelial or mesenchymal identity they have. And so to kind of summarize or give you an idea of what I'm thinking about here, so in a wild-type mesenchymal progenitor, polarity is required to transition this mesenchymal to epithelial cell fate. Polarity is required to, by some means, I believe, very likely indirectly, inhibit PAX to establish this spatial antagonistic relationship between PAX and the WT1 transcription factor to allow renal progenitors to undergo a proper cell state transition. However, when we remove polarity, we do not establish this inhibition of PAX. We do not establish. We fail to maintain proper epithelial identity, and we do not have this the proper establishment of control in the epithelium of the nephron. So there are many lenses through which you can view this. I showed you a very negative lens in cancer. However, in a more positive lens, thinking about this in a more positive way. So again, in the development of a normal nephron, you have this mesenchymal to epithelial cell transition. You form a lumen at the apical center. However, in a more positive light, there's regeneration it's known that the zebrafish is a regenerative monster. It can regenerate almost every organ. And humans don't have this capacity, but we're really trying hard to figure out how to do this in humans. But by some means, this epithelial mesenchymal transition, and numerous labs have found this to be true, explicitly Ian Drummond's lab, the Bonaventure lab in Harvard, have found this to be true, where you can undergo this mesenchymal to epithelial cell transition and undergo this cell state transition to propagate this proper lumen and replenish the cells that are lost. And so to finalize my idea here, again, overall, there's a stemness idea where the overarching ultimate stem cell, the zygote, undergoes this mesenchymal to epithelial cell transition in all germ layers of the zygote, the mesoderm, endoderm, and ectoderm, to form all of your organs of your body. And by some means, we may be able to, reprogramming mechanism whereby iPS cells are undergoing this might have some capacity to undergo this during stem cell biology, and polarity is such a key component of this. I think it's just amazing. For that, I'm happy to call myself a developmental biologist. I think developmental biology is really at the cornerstone of all these ideas. And with that, I would like to thank my lab, and I'll take any questions from the audience, and I appreciate your time here. Thank you very much.